Real quick before this starts, I know I talk really fast, you probably won't be able to understand. Sorry. And I also try and make a joke. It fails because I'm extremely tired when I'm recording this, and it did not go well. So yeah, enjoy if you can. Hi, I'm Brandon. This is Course Crash Biology. Today we're going to talk about mitosis and meiosis. The best way to get a good understanding of mitosis and meiosis is by going through the steps. Let's start with mitosis. The first step is interphase, which is commonly known as the period of growth. There are three separate phases that make up interphase. First, we have G1, which is cell growth. In this phase, the cell grows and synthesizes new proteins and organelles. Next is the S phase. The S stands for synthesis. During this phase, the DNA is synthesized when the chromosomes are replicated. At the end of this phase, the cell has twice as much DNA as before. Lastly, we have the G2 phase. During this phase, many of the organelles and molecules needed for cell division are produced. After this phase, the cell is ready to enter the M phase, or mitosis. The M and N phase comes from mitosis. Once again, there are a few phases, 5 in total, that make up mitosis. They are prophase, metaphase, anaphase, telophase, and cytokinesis. There's only one place to start, prophase. During prophase, the genetic material inside the nucleus condenses and the duplicated chromosomes become visible. Outside the nucleus, a spindle starts to form. The duplicated strands of DNA are attached along their length at an area called the centromere. Each DNA strand in the chromosome is called a chromatid. Once mitosis is complete, the chromatids will have separated and have been divided between the new daughter cells. The spindle fibers that start to form extend a region called the centrosome, where tiny paired structures called centrioles are found. As prophase ends, the chromosomes coil more tightly and the nuclear membrane starts to break down. Then the cell proceeds to the second phase, metaphase. Metaphase is the shortest phase. During metaphase, the centromeres of the chromosomes line up across the center of the cell. Spindle fibers then connect to the centromere of each chromosome to the two poles of the spindle. That's all that happens in metaphase. Next, the cell goes into anaphase. Anaphase is where separation starts to occur. Sister chromatids suddenly separate and begin to move apart. Once anaphase starts, each sister chromatid is considered an individual chromosome. The spindle fibers move the chromosomes to the opposite ends of the cell. Anaphase stops when movement stops and the chromosomes are completely separated into two groups. Then we go to telophase. Telophase is where the cells reform. The chromosomes begin to spread out into a tangle of chromatin, and nuclear envelope reforms around each cluster of chromosomes. The spindle also begins to break apart. Mitosis itself is complete, but there's still one more step, cytokinesis. Cytokinesis tends to occur at the same time as telophase. Cytokinesis completes the process of cell division. It splits one cell into two. During cytokinesis, the cell membrane is drawn inward until the cytoplasm is pinched into two nearly equal parts. Each part contains its own nucleus and organelles. At the end of mitosis, you have two daughter cells that are identical to the parent cell. An example of mitosis in your everyday life is when you get a paper cut. Cells undergo mitosis to repair the cut. Now we have meiosis, which has very similar steps but some differences that are very important. The steps in meiosis are interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis, prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. To make this a little easier to explain, I'm going to group some of the phases together. Once again, just like mitosis, we start with interphase. The cell grows and synthesizes new proteins and organelles. The DNA is duplicated, and many of the organelles and molecules needed for division are produced. Next, we have prophase 1. The cell begins to divide and chromosomes pair up. Each replicated chromosome pairs with its corresponding homologous chromosome. The pair of chromosomes forms a structure called a tetrad. As tetrads are formed, a process called crossing over occurs. The chromosomes cross over each other, then the cross sections are exchanged. It looks a lot like the two chromosomes are shaking hands, but they steal each other's hands. Ha! Got your hand. Oh. You got mine. We'll call it even. As prophase ends, a spindle forms and attaches to each tetrad. Then we move on to metaphase 1 and anaphase 1. During metaphase 1, paired homologous chromosomes line up across the center of the cell. As the cell moves into anaphase, the pairs of chromosomes separate. During anaphase 1, spindle fibers pull each chromosome pair towards the opposite ends of the cell. Once anaphase is complete, the separated chromosomes cluster at opposite ends of the cell. Next is telophase 1 and cytokinesis. During telophase 1, a nuclear membrane forms around each cluster of chromosomes. Cytokinesis follows, forming two new cells. Two daughter cells. We're done, right? Wrong. Unlike mitosis, meiosis creates 4 daughter cells. Let's continue the process. We start what is called meiosis 2 with prophase 2. The only thing that really happens in prophase 2 is the nuclear membrane fragments. No crossing over occurs. Next we have metaphase 2, where the chromosomes once again line up at the center of each cell. In anaphase, the chromosomes are split apart and pulled towards the opposite ends of the cell, again. Then in telophase 2, a nuclear membrane forms around the chromosomes of each cell, and in cytokinesis the cells separate. The result of meiosis is 4 daughter cells with different chromosomes in each, compared to mitosis, which only produces 2 that are identical. The cells produced by meiosis 2 are gametes, or sex cells. That's the process of mitosis and meiosis. Hope you learned something. Thanks for watching, see you next time.